we have discussed the structure of DNA, and last time we have some overviews on DNA replication, some important points. Let's recap that points, right? About DNA replication. In DNA replication, it is must to know that what is replication, what is the origin of replication, right? And what enzymes are responsible for this DNA replication, and what are the drugs on clinical side, what are the drugs which affect this DNA replication during cancer, during viral infection, during bacterial infection, because we all are doctors and we must know the clinical side of this DNA replication, right? DNA replication, the first point which is very basic is, for example, we have two haploid cells. This is womb, which is also known as egg, came from mother, right? And this is sperm came from father, right? This cell have 23 chromosomes, came from mother, which are also known as maternal chromosomes, right? And this sperm have 23 chromosomes, came from father, right? Our life starts even on molecular level, right? Even from one cell. These two cells fertilized. They get fertilized and form a new cell, which is called zygote. But this zygote have 23 chromosomes from here, which is maternal, and 23 chromosomes from here, which is paternal. Right? So now this zygote have 46 chromosomes, 23 from mother and 23 from father. Now this cell will start division. This zygote will start division. For example, it forms two cells, 4, 8, right, and so on, right? But after these mitotic divisions, it form a whole organism. Like for example, this is Rakibunisa form. From that zygote. This is the full body. Right? This is the whole body. Right? And this body is made up of about 100 trillion cells. From where that 100 trillion cells came? From this zygote. Right? For example, these are the cells. Now, full body is made up of cells, about 100 trillion. But all the cells have same genome, same chromosome, same genes, right? For example, these cells have same chromosome or same DNA as hepatocyte. For example, I am taking liver from this body, and also I am taking neuron cell, right? And I am also trying to take stomach cell from this body, right? This stomach is also made up of cells. This is also cell. This is also cell. The cells present here are called hepatocytes, these are called neurons, and these are called gastric cells. Right? But all these cells have the same chromosome or same genetic machinery as in zygote. Now, a question arises here. If all the cells have same DNA, information stores in DNA, then why every cell functions different from each other?
Codons.
That's the question, right? Anyone from you? No?
Genes different, protein synthesis.
Protein
Codons.
synthesis. No? All the cells have same mean. The genes present here, one DNA have about 30,000 genes. 30,000, right? So this, if the DNA is present here, here, same DNA. The genes present here are the genes same present here. I mean that the genes present in neurons are same genes as present in hepatocytes. Then why hepatocyte functions differently from this neuron? This is because of the genes expression, right? For example, I am taking pancreas. This is pancreas, and I am taking a beautiful neuron here. This is a neuron from that world, right? And have two types of cells. One is alpha, second one is beta, right? Here this is neuron. What is the function of neuron? Signal transmission, right? And what is the function of this alpha cell? Glucagon. What is the function of beta cell? Insulin. Right? But this cell and this cell, for example, this is paternal chromosomes in this cell, and these are maternal. Right? Both have same genes. Now, what is the difference between, and also here the genes and the DNA is same as to pancreatic cells, right? Then why they are function differently and why they have different structures and this have different structure? It is because of genes expression. Because of genes expression. For example, I am taking one DNA from alpha cell, right? And I am taking one DNA from beta cell, right? And also I am taking one DNA from neuron. Right? This cell have insulin production gene. This DNA have. It also have insulin productive gene. It also have insulin productive gene. Right? This cell have glucagon production genes. This beta have also. This neuron have also. Right? This DNA have single neurotransmitter production genes here, alpha cell, right? Beta have also neurotransmitters. For example, we are taking serotonin or we are taking acetylcholine. This is the gene for acetylcholine. This is the gene for acetylcholine, and this is also the gene for acetylcholine, right? For example, this is the gene for insulin. This is for glucagon. This is for acetylcholine. Here, this is for acetylcholine. This is for glucagon. This is for insulin. Here, this is for insulin, glucagon, and acetylcholine. Right? All the cells have, all the DNA have these genes. But now, after some time, you will know that there are some special areas for expression. Right? In alpha cell, this gene cannot be expressed. That this gene also cannot express. Only this gene will express and transcription. Right? So this cell is responsible for glucagon production and will function for lowering the glucose level, increasing the glucose level in blood. Right? Here in beta, the gene for insulin only can express. Glucagon gene cannot express, acetylcholine cannot express. So this cell is for insulin production. Right? In neurons, this gene for acetylcholine can express, and these both cannot express in neuron. So neuron is responsible for signal transmission. Right? Because I have already told you that DNA after transcription forms messenger RNA, and messenger RNA forms structural and functional proteins. Right? In alpha cell, for example, the structural and functional proteins formed by this messenger RNA and DNA are different from beta cells. And the structural and functional proteins formed in neurons are different from these both alpha and beta cells. That's why our cells are different in structure and function in body. But all the cells have same DNA. All the cells have same chromosome. Right? Understand?
Yes, sir.
Now, 100 and 1000 times mitosis take place on our body.
Yes.
Right? 1000 and 100 million times lifetime. And all the cells are getting same DNA, same copies. Same copies. Right? For this reason, DNA replication is important. It's necessary. That after cell division, for example, this is parent cell and having 46 chromosomes, right? After one division, it become two cells and getting the same chromosomes and DNA from parent. Now these cells are parent cells and they divides. They divides, right? And also these daughter cells also taking the same genes, the same DNA from parent. For this reason, the DNA replication is necessary and important, is required by body. That all the cells must have same DNA and same gene, right? Now, in DNA replication, you will tell me. You will tell me. You will tell me. What is mitosis? Before mitosis, which phase occurs in body? Interphase. Interphase is divided into three phases. G1 phase, S phase, G2 phase. We have discussed G1 phase in detail. Which things are happens in G1 phase? Proteins. Also, there is some checkpoints,
Yes.
and the genes are responsible for that. The growth factors, the tumor suppressor genes, right? Now, this DNA replication, it's mean that we are discussing S phase, synthetic phase, right? In which DNA copies are formed, right? So let's start DNA replication. Now we will discuss in detail the DNA replication. This is very interesting topic, DNA replication. Fortunately, we are medical students. This is our luck that we are knowing about ourselves, our body, not about our cats and dogs, like zoology students, right?
Yes.
We are known about ourselves. In DNA replication, some important point. There are some important points. Every student must know that points, and we have discussed yesterday a little bit.
Yes, sir.
Right? The first thing, what is replication? What is replication? Simply, it is just making copies. Making copies. So, what is DNA replication? Making the copies of DNA. Right? The second thing, for example, this is our original DNA. This is my original DNA. You know the structure of DNA, right? Now, after replication, this DNA will make two copies. Right? This is called DNA replication. The second, location of or which phase is responsible for this DNA replication? S phase. What is S phase? For example, I have interphase. This is interphase, right? And this is G1 phase, right? This is S phase, and this is G2 phase. This is M, mitosis, right? So this is checkpoint, and this S phase DNA replication occurs. This is the second point which is necessary in S phase, right? The next, third, DNA replication is semi-conservative. DNA replication is semi-conservative. What's mean by semi-conservative? One strand must be old, one new, right? For example, I have this DNA in my cell. This is the parental, or this is the parent DNA. After cell division or after S phase, this DNA will make two copies, but it must have one is blue and one green. That means that one strand is old, original, and this is new copy, right? And this also, this is old and this is new copy. Or it is also known as, this is parent strand, this is daughter strand. This is parent strand, this is daughter strand. This is called semi-conservative because both the strands are complementary to each other but anti-parallel, right? And the new strands forms must bind with one or strands, right? And both have same genes as on parent, right? The next thing, fourth, DNA replication must be bidirectional. It must be bidirectional. Bidirectional means that if I have this DNA replication process, it must be uncoiling in both directions and will replicate in both directions. Right? This is called bidirectional process. Bidirectional, two direction, etc. Right? So this is called bidirectional. The next thing, five. DNA replication occurs 5 to 3. New strand must form in this direction, 5 to 3 end, and the old strand must be in 3 to direction, right? For example, this end is 3 end, and this is 5 end, this is 5 end, and this is 3 end, right? And this DNA have nucleotides. We will discuss this in detail, how this process occurs. This new strand will form in this direction, right? This is 3 and this must be 5, and this 5, this must be 3. So the new strand forms in 5 to 3 directions, and all strand must be in 3 to 5 direction. Right here, this strand will form. Here it is 3, 5. This is 3. So the new strand is 5 to 3 direction, and the old strand is 5 to 3 to 5 direction. Right? This is called the DNA. The replication the new strand must polymerize in 5 to 3 direction. Right? The next thing. DNA strands after and before replication must be complementary and anti-parallel. There is something unjustice to DNA strands, right? We are crying about justice, but there is no justice even on molecular level, right? Because one strand is this DNA making one strand in continuously, right, in continuous pattern, but the second strand forms in fragments. This is injustice with these both strands, right? So this must be complementary and anti-parallel. How? For example, I have this DNA. Complementary means that if here is adenine, here thymine, cytosine, guanine, cytosine, adenine, thymine. Right? Here must be thymine, here must be adenine, here must be guanine, cytosine, guanine, thymine, and adenine. Right? This is complementary. Both the strands are complementary. But this strand is in 3 to 5 direction, and this is in 5 to 3 direction. So both the strands are anti-parallel. Both the strands must be after and before DNA replication. The strands will be in anti-parallel direction, anti-parallel pattern. Right? This is the next point. The third point that a student must know is about the origin of replication. Same one. Origin of replication. Anyone have blue marker? Origin of replication. Right? Origin of replication means there must be a weak point in DNA. Right? Must be before helicase enzyme. Helicase enzymes are responsible for uncoiling, but before helicase enzyme, there is an enzyme which is responsible for separating the two strands. This is just like husband and wife, and someone is trying to separate them. Third party. And the third party also trying to keep them separate for not binding again after separation. Right? So origin of replication is just like that. We have two cells, and we must know about both the prokaryotic and eukaryotic. Right? Here, the origin of replication is the area from where the replication starts. This area is also known as consensus, and it is also known as AT-rich area. Right? So, for example, the origin of replication must be weak. Weak means having double hydrogen bond, right? And this property is present in AT-rich So this area of replication is also known as AT-rich area, or it also known as consensus. What is the spelling of consensus? Right? Consensus. Check it and correct it. Right? But that is also known as consensus. Right? And it is also known as enriched area. Right? It is after separation. It is also known as replication bubble. Replication bubble. Right? For example, I have DNA here. This, for example, DNA. Right? Originally, this DNA is like this. Right? Someone is holding this strand, and from here it also holding this strand and trying to separate these two strands, and they are now successful and separated. Right? This area is called origin of replication, because the replication starts from here. Right? Replication starts from here. Right? The next thing, it is also known as replication bubble. But now we have two cells. For example, I am taking both, because when we are using drugs, we are applying their drugs on bacteria, virus, etc. And we are trying to stop or inhibit the DNA replication and that bacteria or virus. But if we are caused by cancer, we are trying to stop this DNA replication in our eukaryotic cell. Right? So, for example, I have two cells. This is prokaryotic. For example, this is bacteria, and it is E. coli bacteria. Right? And this have a DNA molecule here. This is double-stranded DNA present in bacteria, E. coli. Right? And for example, I have human cell, eukaryotic cell. Of course, it will be round, but it has a long DNA. You know what is the length of DNA in one cell? Anyone?
2 nanometers.
What is the length of DNA? Is 1 meter long. The length of DNA, one DNA is 1 meter. Right? There is something very long present in the cell. A cell is very minute particle. Right? So this is elongated DNA present in eukaryotic cell. Right? But the difference is this DNA has only one origin of replication. Right? These enzymes are trying to separate these two. Right? And this DNA will make two another strand. For example, this is one strand and this is another. And now, when after binary fission, for example, this bacteria replicates in two cells, because there is no mitosis required in bacteria. You know, no mitosis is required in bacteria. Why? Why?
Equal number of division.
Dr. Saab. Why bacteria cell have no need of mitosis and we have the need of mitosis? It is unicellular and can easily divide by binary fission or fragmentation. Right? So after binary fission, when the cell splits into two, before this it must pass from interphase, DNA replication. Right? So now from here, for example, this is replicated DNA. One strand is One strand is new. This blue is new, and this red strand is old. This is one DNA formed from this, and this is another. This is original strand, and this is copied strand. Right? But the point to remember here that bacterial cell or prokaryotic cells DNA have only one origin of replication. Only one origin of replication. As compared to prokaryotic cell, if we take eukaryotic cell or human cell, it may have more area of replication. For example, I am taking this DNA here. For example, it can form a bubble here. It can form a bubble here. And it can form a bubble here. Right? So, this eukaryotic DNA have more than one origin of replication, and this DNA have only one origin of replication. Right? When one bubble found in eukaryotic DNA, the whole DNA must have to replicate. Right? And this DNA proteins came and separate these strands from each other. This DNA is tend to replicate. Right? What are these areas? These are AT-rich areas. These are AT-rich areas, and these are called origins of replication. Origin of replication. Origin of replication. So the point here is, in prokaryotic cell there is one origin of replication, and eukaryotic cell it must have more than one origin of replication. This is the next point. Right? The third one, the DNA, the replication forks, formation of دي بس ربيع وزيتين ها بس سرب زمان في كلاس
وين تستاسو كلاس مو رازو
سيدة كمان سرب
صار
زمان
نبيعهم تنخلو
You will sit on this side and the males will sit on that side. सही है? आप इधर बैठेंगे, वो उधर बैठेंगे. हाँ. So this is the next point, that replication bubble or origin of replication. And eukaryotic cell they have a lot of origin of replication, and prokaryotic cell only one replication bubble is here. Right? The next point, the replication fork. 9. Replication fork. Right? For example, I have a replication bubble, or this is the origin of replication. This area from where the DNA is uncoiled, this area is called replication fork. Right? One bubble have how many replication forks? Two. Every bubble have two. One on each side. Right? The next thing. Next. The replication must be in DNA replication must be in whole DNA. Right? I have already told. When one replication bubble arises, all the DNA must have to replicate. Right? But this replication process is very careful process. It's very careful. It have no mistakes, or it have very less mistakes. And there are some enzymes which are very beautiful and very intelligent, they have three eyes. Enzymes have three eyes. Two on this side and one on this side. Right? And they are circulating in DNA. And every time scanning the DNA, every time scanning the DNA. Right? And when they are repairing DNA, also proofread their DNA from this side. The work I have done, it is wrong or right. Nasi kana, jadi bagaimana kerawani ya enzyme ne, ruste bewayu. Nah, dia makin gurih, dia ganti guna nak kerja no enzyme. Sekali ganti kali dia akan cut kinor pakai lagi, nucleotide. Oh, pakpal itu pakpal karu kita seperti ini terus buat apa dia sekarang gurih cuma ada karu udah tik tik galat. Right? So this process have no mistakes or very less mistakes, right? But the enzyme, that beautiful enzyme, we can frank with that enzyme, and we can treat some diseases by that enzyme also. Right?
Yes.
We will discuss that in detail later. Right? So these are the important points about DNA replication. The next point: DNA replication must occur in three steps. Having three steps. The first step is initiation. यहाँ से DNA replication शुरू होती है. The next is elongation phase, and the third one is termination phase. Right? This is the next point in DNA replication. We will start from here. Right? The next thing, 12th point is enzymes and proteins. Enzymes used in DNA replication. I am trying to tell you some names, some important names. Right? The first one is, the important proteins are pro-replication proteins. Right? The next is single strand binder proteins. They are single strand binders, single strand binding proteins. Right? The next is DNA helicase. The next is DNA dependent DNA polymerase. Right? The next one is DNA dependent अब क्या करें हम?
यही करना है कि कोशिश करें पीछे कुछ और करके गुजर चले जाएंगे कुछ हो जाएगा
ठीक है नंदन तेरा सब क्लास खत्म है
ना वो क्लास खत्म हो आफ्टर ब्रेक हो गया जब बाद क्लास खत्म
आओ
सही
सही वापस से फुल कंबाइन करें
ये हमारा गेची
सर पर
इंस्पेक्शन
आओ
बाकी में कंबाइन
DNA dependent. Attention please. Attention please. DNA dependent DNA polymerase. The next one is DNA dependent RNA polymerase. RNA polymerase. But there are some other names also for these two DNA polymerase. But DNA polymerase have a lot of types, and they are very keen. They are very clever, right? They are very clever. These are called RNA primase. You have idea about RNA primase and DNA polymerase because I have given you, right? The next is topoisomerase. And topoisomerase have also types 1, 2, 3, but they are structurally different. Functionally also they are different.
Yes.
Right? The one and only enzyme which are using ATP in very abundant amount is DNA helicase. It used ATP in abundantly. It was a lot of use of DNA, ATP. Sorry, right? Topoisomerase are also very important, and we can block these enzymes. Right? The next is the last one is, which is very beautiful, having beautiful oils, is telomerase enzymes. Right? They terminate the process. Telomerase. Right? And then these are the enzymes we were discussing DNA replication in very big detail. There's about 2 hours lecture on DNA replication, including this. Right? But these are the basics. These are some crystal concepts about DNA replication, and now we will go for DNA replication in detail. That how the raw material comes from cytoplasm to nucleus or from nucleoplasm, and they are making DNA strands, and how the DNA strands are read and proofread. There is 2 types of reading. One is reading, simple scanning. The second is proofreading. Proofreading means that I am, for example, an enzyme is working and he confirms that the work I have did is right or wrong. This is proofreading, and this is the beauty of these enzymes. No one enzyme in body can proofread your work except DNA replication enzymes. Right? समझ गए?
Yes, sir.
हाँ. Now,